overview about sterile filtration in the biopharmaceutical manufacturing. I prepared this presentation, want to give a technical overview, also starting with basics and then more and more going to more detail, showing some trends in the market and tell you something about regulatory requirements. So the content for today, I'm currently in the introduction phase, prepared something about filtration basics, what are retention mechanisms, membrane materials, what is currently being available on the market, what are the trends when we talk about materials, what kind of device formats do we have to talk about, filter construction, so how is a filter constructed and what is really important to know, for example, when it comes to dead volumes and so on, so when you want to get highest yield for your process. Single-use manufacturing, some specialties, also about filtration. And then finally the topic filter integrity testing. And there's a big discussion ongoing about PUPSIT, pre-use post-sterilization integrity testing. So I want to give you the latest update here and finally some upcoming trends. But first of all, let me briefly introduce myself. So my name is Jens Meyer. I'm working with Sterile and Biotech since 13 years now. And those of you who are not so familiar with that company, so we are number 3 in filtration, number 1 in fluid management, and number 1 in fermentation globally. We see filtration technologies and the membrane casting procedure as one of our core competencies. And that's the reason why I think we are a really good partner for discussing this kind of topics. Talking about the history, it's already 100 years ago. So in 1918, Professor Zsigmondy in Göttingen invented the membrane filter. So it seems to be a rather old technology, but still very valid and up to date because there's nothing detectable on the horizon which could replace sterilizing grade membrane filters by other technologies. Why is this starting point so important? So as you know, I'm from Sartorius company, and at that time, beginning of the last century, Professor Zsigmondy worked together with our company founder, Florence Sartorius, creating the first and founding the first company selling membrane filters into the market. But anyhow, it took a couple of years, and starting in the 1930s, the membrane filters were firstly used in the pharmaceutical industry. But at that time, not this kind of cartridge format and capsule format we all know, but mainly flat filters in large diameter. So for example, 293 millimeter flat disc filters at that time. And it took 40 more years until finally these kind of pleated filter elements were introduced in the market, and also to get the 0.2 micron rated filters, sterilizing grade filters, becoming the standard in the industry. Until that time, people were happy to retain at least some dirt, etc. But more and more, it was obvious that the sterilizing grade capability to retain bacteria reliably, it's more and more into the focus of the regulatory authorities. In the 80s, the first heterogeneous double layer filters were introduced into the market. I will touch this point later, what this means. It has tremendous impact on the performance of those filters. And at the end of the 80s, also asymmetric membranes were introduced. Also some details, background details later during the talk. In the 90s, the asymmetric polyether sulfone material was introduced into the market as a high performance membrane. And that's currently state-of-the-art filter material when we talk about sterilizing grade filters, but also about pre-filters. So all the latest product launches in filtration is based on polyether sulfone membrane material. In the 2000s, there were some new pore size combinations showing that the pre-filter porosity has a tremendous impact on the overall performance of the filter. Also here's some data later during this talk. And single-use manufacturing from that time on was really focused in many, many companies with concerns, but also with great hope to solve some issues in biopharmaceutical manufacturing. And in the 2010s, there were some minor innovations on new pleating technology, new surface modification to get less adsorptive filter and so on. And the trend not only to have filters in the market, but to have complete single-use unit operations. That's indeed more than just a filter. That was really the trend in the market, and this is currently ongoing with some new tasks that we as filter suppliers have, of course, to fulfill. So over the years, this was a trend towards safe manufacturing process, more capacity, so it's trend towards process optimization, and also cleanliness gets more and more into focus. So the avoidance of particles being released, but also when we talk about extractables and leachables being released from plastic components cannot be completely avoided, but a clear trend to reduce this kind of leachable substances into the liquid. So the basic question for us is what has to be removed from the liquid? It's clear, microorganisms. When we talk about contaminations, if it's an upstream step like media filtration, of course, we want to avoid that a different organism will ever have the chance to enter the fermenter. So all kinds of bacteria and yeast should be retained reliably. But there's more of that. So also particles that may be generated during the process using different process steps should be reliably retained. And this is, of course, highly important and critical for the final filtration step right before filling. The same for aggregates. So we cannot fully exclude the generation of protein aggregates during the process, but it has to be assured that those aggregates will be reliably removed from the product by filtration technologies or other technologies being offered by the industry. So talking about the word of filtration, so where are we really when we talk about sterilizing grade filters? So as you know, a sterilizing grade filter is rated as a 0.2 or 0.22 micron pore size. So this simply means this is the range here that is indicated with this red bar. We talk this about, we call this microfiltration. So all particles being larger than the pore size of 0.2 will be reliably retained, including blood cells, but even small or larger organisms, yeast, bacteria, and so on. When it comes to smaller organisms, and there may be also some specific bacteria like mycoplasma, viruses, monoclonal antibodies, but also small, larger molecules, smaller molecules, then of course we have to apply different technologies. So this is a really small filtration step within the whole range of different particles and molecules and living organisms to be retained from any liquid. So how is it done? What is the working principle of a liquid filter? It's very simple. So it's a pore structure within the membrane. But it's very important not only to think about single pores within the foil, for example, but it's a more sponge-like structure. So a complex three-dimensional arrangement of cavities where any kind of particles, including bacteria and other organisms, have to go through for ever having a chance to get access to the other side of the membrane and to be released again. And this is really complicated. And for such small organisms like bacteria here, you see it's not a pore with an ideal pore size, so you cannot really measure it and you will always get 0.2, but it's more a statistical approach. And how a sterilizing grade filter is qualified, I will touch this point in a few minutes. So very important, this is a picture, a real picture of a membrane, of a polyether sulfone membrane, challenged with some microorganisms, Brevondomonas diminuta, during a test that we call bacterial challenge test. And this is exactly the burden we put on such a membrane, and only when it is guaranteed that not a single of those microorganisms will ever have a chance to be released from the downstream side of the filter, then we as a supplier are allowed to call this membrane is sterilizing grade filtration membrane, and the product is sterilizing grade filter. So this is really important to understand. And of course, you can see that the size of the microorganisms is very close to the pore size of the membrane. And again, it's not only the pore that is lying on the upper surface of the membrane, but it's a really complex three-dimensional structure. And most of the organisms of the bacteria will be retained at the surface, but some will have maybe a chance to get access to the first pores, but then will be finally retained within the matrix of the membrane, and not a single bacterium will ever have a chance to be released and potentially causing contamination into the liquid. So the retention is done first by size exclusion, so very important, so that the pores have to be smaller than the particles to be filtered. And that's clear that the larger particles then will be automatically mechanically retained by this structure. But also smaller particles can be retained, and then we talk about absorption. So this is finally a kind of surface interaction. When there's a surface charge on the particle and an opposite surface charge on the membrane, they will be attracted and will be captured by the membrane itself. So it's not only size exclusion, but it's also pretty much absorption that leads to reliable retention of smaller particles compared to the pore size of the membrane. So what pore sizes are relevant in the pharmaceutical industry? We are talking about pre-filtration when the pore size of the membrane is larger than 0.2 micron. So then we talk about bioburden reduction, and this filter is used as a particle filter, also for protecting the sterilizing grade filter, which is more expensive and has a higher value. The real sterilizing grade filtration step is rated as 0.2 micron membrane. And that's independent which material is used. So there are different membrane materials being available on the market, and always the filter suppliers have to assure that these criteria are fulfilled. How this is qualified, give me a few minutes more to explain this. But there are also smaller membranes being available, especially for mycoplasma retention. So mycoplasma is a specific microorganism, a bacteria, which is known to successfully pass also sterilizing grade filter membranes, and then finally leading to contamination of the bioreactor content. So in case that you have some risk for mycoplasma contamination of your incoming goods, then of course such a mycoplasma retentive filter is of high value for your media filtration step, for example. And even smaller when it comes to virus filtration, it's 0.02 microns. But always you have to keep in mind that the smaller the pore sizes get, of course, the higher the safety is. But at the same side, the flow rate goes down. So the performance-wise, of course, you have less flow rate, maybe also earlier blockage of the filter, but increased safety. And even smaller pore sizes, and we are in the range of ultra and nanofiltration. So these are cross-flow diafiltration applications where it comes really to molecular separation. But for today, we are concentrating more on sterilizing grade filtration step. So sterilizing grade filtration step is kind of static filtration, or also called dead-end filtration. Why is this? Because the liquid comes from above, penetrates the membrane, and will be released at the downstream side of the membrane. And the particles will be retained by the membrane and laid down on the surface. This offers, of course, the risk when you have a high particle burden that the membrane gets more and more blocked the more liquid you filter. On the other side, we have a technology called cross-flow filtration. I will just briefly touch this. It's a different technology. So you have two membranes. The liquid passes the membrane in an orthogonal direction. Liquid will pass the membrane, but you have always a constant flow directly below the membrane. And by this, you actively remove any particles being retained from the membrane, so you will avoid this kind of early blockage from the membrane itself. This technology is mainly used for cross-flow applications, And when you remember the previous slide, it has something to do with molecular separation, where you want to diafiltrate and concentrate your liquid to a certain extent. And then, of course, you have to avoid a blockage by the retained particles. And therefore, this kind of cross-flow filtration is beneficial. But for today, we are concentrating only on this kind of static dead-end filtration. And even when we talk about this kind of dead-end filtration, we have to distinguish between fleece filters, fleece-based filters, and membrane filters. So fleece-based filters is really a kind of fleece that are arranged in a three-dimensional mesh. And depending on the complexity and on the density of the different fibers being put together, you will get a certain nominal retention, how it's called. So this kind of fleece filter can be polypropylene filter, glass fiber filters, is mainly used for pre-filtration and clarification steps, and particle deposition. So as a pre-filtration, when you want to get rid of high particle load, then this kind of pre-filtration step based on fleece filter technology is really of highest value. Why this? Because you have a really high dirt holding capacity. So you can produce this kind of fleece in a with a very high thickness, and by that you have a really high dirt holding capacity. On the other side, you have the membrane filters. The membrane filters is, when we talk about sterilizing grade filtration, it's always a membrane filter. And why this? The main advantage is simply you have absolute retention compared to the nominal retention of the fleece filter. So these membrane filters can also be used for bioburden reduction. So when you have a certain bioburden that you want to reduce, but not necessarily down to sterilizing grade, then you can use something which is has a pore size of more than or greater than 0.2 microns. But also used for sterile filtration, so 0.2 micron, for mycoplasma filtration, 0.1, and also for virus filtration and nanofiltration. These kind of fleece filters, why are they limited? This is, of course, an important question. As I mentioned, you have a really high dirt holding capacity. You have a retention via sieve effect, so it's mechanically retained, and also adsorption, so the same principle, underlying principles as for membrane filtration, but you have only a nominal retention. And why this? Simply because when you have low pressure, the particles will be retained by the three-dimensional mesh, but not only at the surface, but the particles, especially the smaller particles, will get access or will be retained in the depth of the fleece. And when you have, for example, higher pressure or pressure pulsation during your process, there is a certain risk that single particles will be released by the downstream side of the filter. And this is, of course, something which is not acceptable when we talk about sterilizing grade filtration, because then nothing is allowed, not a single bacterium is allowed to pass through it. So typical materials for these kind of fleece filters is glass fiber, polypropylene, and cellulose. And depending on the chemical resistance and degree of adsorption, you can choose between different materials. And you can see here, so glass fiber is allowed to be used in pH 1 to 10, polypropylene in the full pH range 1 to 14, and cellulose is a little bit limited between 4 to 8, but for most of the biopharmaceutical applications, this is not a big deal. In contrast to the fleece-based filters, the membrane-based filters, as I mentioned, they have this kind of homogeneous sponge-like pore structure with a very high mechanical stability and pressure resistance. So the main advantages are you have an absolute retention. This creates highest safety. So when you have a validated sterilizing grade filter, you can be sure that under standard process conditions for the single bacterium will be released. And another very important difference to the fleece-based filters, they are testable. So integrity tests, filter integrity tests can be performed before and after use to increase the safety even more. They are mechanically robust, so they will accept this kind of pressure pulsations and so on. But there are also some disadvantages. And this is simply the low dirt holding capacity. So all the particles, in contrast to the fleece-based filters, are retained directly at the surface of the membrane. And then you can imagine that when you have a high particle load, more and more particles will settle down to the membrane, leading to a certain degree of filter blockage. So this simply may lead to early filter blocking, and is a good reason why for some processes it could make sense to position a pre-filter in front of the final filter. So there are different membrane materials being available on the market. So the most relevant one for today is polyether sulfone, PES. It's characterized by a very high capacity and flow rate due to the asymmetrical membrane structure. I will show some pictures on the next slide. Another material is PVDF. It's a rather old material, since 30, 40 years being available in the market. In contrast to polyether sulfone, it has a relatively low capacity, so it tends to block earlier compared to the polyether sulfone membrane, and also lower flow rate, but it's characterized by low absorption. So this absorption compared to PES is much lower. So for example, when it comes to a final filtration step, there is a certain advantage of PVDF material compared to PES. The same is true for cellulose acetate. It's a different material, medium capacity flow, but here also the key feature is low absorption. So feature-wise, PVDF and cellulose acetate are very similar, and polyether sulfone for all processes that are really have a clear demand for high capacity, where certain filter blockage may occur. Finally, we have polyamide, also called nylon. We have high absorption here, but here the main benefit is the high chemical resistance. For example, when it comes to antibiotic processes, with a lot of solvents being used, then this kind of nylon filters are superior to the other one. And so you see, depending on the need you have for your filtration process, you can select one of those different materials. For all the materials, of course, it has to be guaranteed that sterile and grade filtration is done. I briefly mentioned the different symmetrical and asymmetrical membranes. Symmetrical membrane, this is valid for polyamide, cellulose acetate, and PVDF. This simply means that the pore size is the same across the entire membrane. So you start with a pore size, let me call it 0.2 at the top of the membrane, and it continues like this until the downstream side of the membrane. Asymmetrical membranes, and this is polyether sulfone mainly. You can see already here in this scanning electron microscopic picture, the pore size of the pores close to the upper surface are much higher compared to the downstream side of the filter. So there's a certain pore size gradient. The pore size simply gets smaller the deeper you go into the membrane. So why is it so important? So it's simply it's symbolized by this. It's a funnel-like structure, and you get a higher throughput by fractionated separation. What does it mean? It simply means that the larger molecules are retained at the surface, and the smaller the molecule, the particles get, the deeper they get access to the inner matrix. And by this, you do not only use the upper surface of the membrane for retention, but you use more of the material, and this leading to a higher capacity of the filter. So this simply means these kind of asymmetric filter material, membrane material, leads to a higher total throughput capacity. Simply, you only need less filters for a given process compared to the symmetric membranes. So here you can see the membrane, the molecules being retained at all the particles being retained at the surface, leading to early blockage. And this can be really a big difference in a given process. And it is possible to modify even the asymmetric polyether sulfone membrane in a different way, so that you get small degree of asymmetry and a larger degree of asymmetry. And this depends on the need for the filter you want to see. when it's, for example, a pre-filter step, and you have a broad particle spectrum in your liquid. For example, after cell harvest, where you have all the cell debris and the molecules and so on, then you will get this kind of structure, the high asymmetric structure, which offers a much bigger dirt holding capacity than the smaller one. But anyhow, when it comes to sterilizing grade filters, it's not only the dirt holding capacity, it's more importantly the bacterial retentive characteristics. So a sterilizing grade filter, as I explained at the beginning, is not allowed to release any bacteria during the filtration process. And therefore, when the degree of asymmetry is like that, this is mainly the membrane which is used for retaining the bacteria and for guaranteeing the sterilizing grade characteristics of the filter itself. So the degree can be adjusted, defined by the membrane casting process, and that's of course the key know-how of the filter suppliers. They can be adjusted for different needs, and there are different needs depending on the processes and the process steps we're talking about. And this also explains the different performance of PES filters. So there are several different PES filters being available in the market, and they are not directly comparable performance-wise, but there are big differences. And that's finally the background information for the explanation. At the beginning, I introduced also the heterogeneous double layer concept. It's a kind of built-in pre-filter. So how is it done? It's not only one membrane being used for building such a filter, but two membranes. So here you can see two layers, one pre-filter layer. This is an example here of a 0.45 micron rated filter, followed by the final filter, 0.2. And it's a similar story as I just explained with the asymmetric membrane. Why do we use this kind of heterogeneous double layer? Simply because the first layer is there for protecting the final membrane. So when it comes to particles in the liquid, the first layer will retain all the big particles, and by this protecting the final filter membrane, which is the 0.2 sterilizing grade filtration membrane. And why is it so important? It's simply because when you have only a single layer membrane, all these particles, the bigger particles, would directly get access to the final filter membrane, and thus leading to early blockage of the whole filter. So this kind of double layer concept led to enormous increase of the total filter capacity, and by this it's possible to reduce the amount of filters needed for a given process significantly. But nowadays, this is standard to use this kind of heterogeneous double layer concept. Here's a picture how it is working. So this is two membranes, the pre-filter membrane 0.45 and the final filter membrane 0.2. And here you can see all the dirt, all the particles were really successfully retained already by the pre-filter layer, and the final filter layer is perfectly white and getting perfectly protected by the 0.45 filter. And by this, the whole arrangement of a double heterogeneous double layer was really working very efficiently. So the first membrane layer protects the second layer from early blockage. That's the story about the heterogeneous double layer. So when we talk about filter construction, it's now like this. You have two membranes, so the double layer construction I just explained, for example, a 0.45 membrane followed by a 0.2 micron membrane, and then you need certain drainage layer. So in most cases, it's polypropylene or polyester fleece, because when you have a pleated device with this kind of zigzag structure of the membrane, you have to make sure that the liquid gets access to the entire membrane. And when you have a very dense packaging of these kind of membrane pleats in a cartridge or capsule, it may happen that the membrane directly press with a specific force to each other so that the liquid has no chance to enter. So this kind of drainage layer has not so much a active filtration role, but more a the function to guarantee direct access of the entire membrane. How is it done in the filter cartridge? Here's a cross section of the filter cartridge, so you can see the zigzag structure, what we call a pleated membrane. So this is a cartridge, you know how it's looking like. When you open it, you get really like a big surface of membrane being implemented in this kind of filtration cartridge. Important to know that the direction of filtration is always from the outside to the inside. So you start here from the outside, so through the outer cage, you have to pass, or the liquid has to pass first the layer of polypropylene, and then the first layer 0.45, then the second layer 0.2, and then another polypropylene layer, and the sterilized liquid will be collected in the inner core and then can be used for it can be pushed into bags or vessels wherever it will be stored or further processed. Very important always is the information filtration from the outside to inside. Why is it so important? It's simply because the construction of the filter is like that. Like an egg, you can put enormous pressure from the outside and the pleating package will be very, very stable. When you try the same from the inside, when you put some forces to the inner structure of the pleat, you act against the stability of the pleating package. And by this, you can destroy the complete filter. And it's the same for an egg. When you try to open it from the inside, it's very, very easy. And that's the reason why the small chickens can escape from the egg when they're hatching. So here's now an example of a membrane. When we talk about membrane geometry, I explained the heterogeneous double layer. I explained the asymmetric structure of the membrane. And this is now here for a real product, which we call Sandle 2XLM. This is the first layer of the membrane, has a thickness of 160 micron, plus minus. It's a 0.2 micron, highly asymmetric PS membrane. It's the first layer, and as I mentioned before, it has a function to protect the final layer. So highly asymmetric. It's followed by a 0.1 micron. It's a less asymmetric PS membrane. And why this? Because simply the second layer has a function to retain the bacteria that may have a chance to pass the first layer. But this will be only a very few bacteria. But this is the reason why we have a less asymmetric structure, because we have less particle burden, just the function to retain potentially existing bacteria. So when you have this kind of cross-section through the membrane, you start with the first layer with very large pores, and then you have this gradient, so it's getting smaller and smaller. And very important, when you have the second layer, it has directly to be adjusted to the first layer. Then this will result in highest performance of the whole filter. The second layer then will be has only the function to ensure sterility of the filtrate. So important to understand the pore size gradient and the perfect match between the pre and final membrane for getting best filtration results. And that's the reason why there are different filters in the market. Not every filter is comparable, and you have some liquids or applications where the one filter fits better than the other one. So this has to be really tested in real life tests. Talking about the membrane, now you know how the membrane is brought into the filter cartridge or capsule. It's also very important to understand that from the outside, when you have a look to the filter cartridges, to a 10-inch filter cartridge, they always look very similar. But there can be huge differences between them. So simply here comparing the filtration area, the effective filtration area per filter, you find something in the market between 0.5, 0.6, 0.8, and even 1.0 square meter per 10-inch. What does it mean? It simply means that when you step from 0.5 to 0.6, it's already 20% more. 0.8, it's already 60% more, and 1 square meter is double the membrane area compared to 0.5. And then it's very important that the total throughput capacity, there's a linear relationship between the capacity and the membrane area. So this simply means that you need less filters of the high membrane area cartridge compared to the cartridge with a lower membrane area. And that's important to know, because when you compare the different data sheets of different suppliers and filter types, you always have to keep in mind what is inside. It's not only PES, that's easy, but just make sure that you get a better understanding what is really the effective membrane area inside such a filter cartridge. Because when you have 30% more membrane, this will result in 1/3 less filter consumption. This can be significant over the years. This can be sure. So higher filtration area, it's less filters needed. And then when we talk about filter formats, we have both. We have filter cartridges, so single cartridges. Those cartridges have to be put into a stainless steel holder. So this can be single filter cartridge or this kind of multi-round for really big applications. And the same kind of formats are available now also for single-use applications, so in capsule format, so self-contained filter capsules, different sizes. So this is the smallest pleated filter element with 150 square centimeter only, up to 30-inch maxi cups, which have 3 square meters. So you see already we have a huge spectrum of different membrane areas available to fit really the membrane area to the process needs. Very important to understand is that we have, of course, different applications and different key requirements for a filter. When we talk, for example, about applications like buffer filtration, buffer filtration is an easy job. So most of the buffers are water-based and not blocking, and this kind of process utilities, the requirement is simply the flow rate. When you have a huge tank of buffer, of course, you want to get it filtered within a certain time. And you're looking for an economic solution. There is no need for many buffers, at least, to get it sterile filtered and so on. So that's different when we talk about cell culture media and downstream intermediates. So here, of course, the capacity is really highly relevant, because cell culture media may tend to block the filter membrane in contrast to buffers. So here, when you see a certain risk of blocking behavior, you need more filter cartridges or capsules compared to this kind of buffer filtration step. And moreover, it's not only the capacity that is relevant, but also when you have a filtration step of cell culture media, you have to make sure that you will not negatively impact cell growth and productivity of the process, and of course. And the more it goes into the direction of the filling needle, so bulk drug substance and drug product, there are different needs. Low absorption. You want to increase the yield. You want to see your final molecule in the vial and not sticking to the membrane, of course. So low absorption is a key issue. Lowest extractables and leachables. So nothing should be released to your final product, but your molecule should be inside. And highest cleanliness, of course, also when it comes to particles and everything which is not should not be in the filtrate afterwards. So you see, this is really the reason why still we have different materials in the market, because there's not the one and only material that fulfills all the requirements. We have high performance filters, we have low absorptive filters, and you have to select the best material for your certain process step. So when it comes to selection criteria to choose the right filter for a process, what is really the parameter that we are interested in? First of all, it's the flow rate, of course. So it's important if there are only droplets getting out of the sterile filter of the filter, or if there's a constant flow. The total throughput, the capacity, it simply tells us how many cartridges or capsules are used for a given batch. The chemical compatibility. So in most filtration steps, of course, that's not highly relevant, but when it comes to solvent filtration and for example, ADC processes where some solvents used in the intermediate stages, this can be really relevant. Safety, of course, is a need. So you have to guarantee sterility, and therefore you have to identify potential ingredients in your product that may interfere with the sterilizing rate behavior of the cartridge. Yield loss. So I mentioned already absorption. This can be significant, and there may be significant differences in absorption when we compare different materials. And yield loss can also be the construction of the filter when it comes to that volume. There is a difference, of course, when there is 100 milliliter being retained from the capsule and not getting out, or if it's only a few ml. When we have a really high value product, this makes really significant difference. And as I mentioned before, cell growth is also something that should be mentioned. This all leads to filtration costs. What are filtration costs? It's not only the filter price. As I mentioned, compare what is really the membrane area inside such a cartridge. And that's the first exercise we all should do. This will tell us also something about the numbers of filters per batch. So what is the capacity of the filter? It's not so easy to get to the information. This you will not find in any data sheet, but here we have to concentrate on real trials, compare different membrane material, and then we will get afterwards results, what material and what filter performs best for your process. Filtration time, of course, the flow rate, time is money. Water consumption, the wettability of the membranes can differ significantly. And don't forget, you have to wet your filter reliably before integrity testing. And this can be a huge amount of water being used there. And this can, of course, make a difference because we are not talking about cheap water, but in most cases about WFI with a certain value. Reliable integrity testing, most of the complaints that we get from the field when it comes to integrity testing is due to insufficient wetting of the cartridge. So because it was not long enough or not enough pressure difference was used for the wetting procedure. So the easier the membrane is wettable, the best for the process and for the operator. The less trouble you will see afterwards. Ease of use. So how is the handling of the filter? How can it be brought into a complex single-use assembly? How can the information about the filter being transferred to the system? Can I use the barcode scanner, for example, to get the lot number and the individual number and so on? Documentation, so what is offered by the suppliers? How will this documentation help me when it comes to regulatory filings? Yield loss. What is the absorption? Is there a certain product loss that I have to accept because my membrane is highly absorptive? The cleanliness of the filter, leachables, extractables. So what is really the difference here? And is there some information being available? Cleaning validation versus single use, and so on and so on. And so finally, the filtration cost is not only the purchase price. This accounts for something like 20% of the total filtration cost. But most of the filtration costs are so-called hidden costs. And here is a huge optimization potential that we see in the market, and where technical specialists can help the users to gain some value just by changing process parameters of different parts of the equipment. So filtration costs defined by various factors. And just an example here. So here you can see the absorptive behavior of the membrane. So it's always a 10-inch cartridge was tested, and here is the protein binding gram per square meter filtration area. You can see it's a great variability between 0.1 and 0.78. So there's 700 milligram difference. And depending on the final value of your target protein, this may easily exceed the purchasing price of the whole filter. So of course it makes sense to check the absorptive behavior of a plastic component before validating this kind of product into a process. I mentioned the single layer, double layer construction, yeah? Just as a reminder. So 0.2 as a single layer, early blockage, and 0.45, 0.2, the first layer retains the particles, and second layer ensures sterility. But when it comes to flow rate, you have two membranes, so two resistances to overcome. So the single layer filter will always show a higher flow rate. So when I come back to the buffer filtration, it makes pretty much sense to use a single layer filter for buffers, for non-blocking buffers, because you do not need the first layer. And by this, you get a higher flow rate and can make your buffer filtration in a much shorter time compared to the double layer filter. So 30% more flow rate is easily possible. So here's an example, different filters. The first one is the single layer construction compared to the different filters with a double layer construction. And you see already between those filters from our own Sandle range, there's 1/3 difference. So it makes sense, of course, to think about what is really the demand of the filtration step and then carefully select the best filter for this process. But of course, when it comes to the total throughput, the first layer has an advantage because it retains the particles already. And the single layer membrane will tend to show early blocking. So here another example, total throughput, so the capacity of a filter, the Sandle 2HS, the single layer version, which was outperforming all the others flow rate-wise in a water study, it's really down on the floor, and there's a difference of a factor of 4.5 in this example, which outperforms the heterogeneous double layer construction just by this inbuilt pre-filter function. So again, when there's a non-blocking filtration step like buffer water filtration, I would recommend the single layer construction for getting highest flow. As soon as there is a certain particle load that may offer a risk for early blocking, then a heterogeneous double layer construction makes sense because the capacity is significantly increased, and by this you need less filters for a given branch. So this, of course, has to be identified and verified in this kind of small-scale trials. So the chemical compatibility can be tested, absorptive behavior can be tested, the identification of the best membrane material is done, what is the optimal pore size combination, is it really 0.45, 0.2, or could there a finer pre-filter beneficial or not? Is a pre-filter required, yes or no? And if yes, which is the best one? So a lot of questions, and these can only be addressed and answered by database and by this kind of trial work. You can put a small scale filter, filter your specific product, and you will get customized reports what really the membrane, what membrane and what membrane combination will outperform all the others. As I mentioned before, the standard is in the industry is 0.45, 0.2 for a heterogeneous double layer. But a couple of years ago, we identified that we are not only filtering one product. And it makes sense, of course, to play a little bit with this pre-filter membrane, because as I explained, the pre-filter layer has only the function to retain particles. And depending on the specific size of the particles in the liquid, it makes sense to use coarser pre-filter membranes or finer pre-filter membranes. The sterilizing grade filter membrane is always identical 0.2. And just one example here. So again, this is a double layer construction, different products. The same product was filtered and afterwards opened, and just photos were taken, and you easily see that this construction here, 0.8, 0.2, you see a lot of dirt on the 0.2 micron membrane. This finally led to a premature blocking, so the capacity was less and you need more filtration area, more cartridges or more capsules for a given process. The 0.45, 0.2 was better, so a lot of dirt was already retained by the 0.45 in contrast to the 0.8, and the 0.2 was much better protected compared to that example here. The best example here was the 0.35, 0.2, where most of the dirt was retained by the first layer, and the final layer was more or less clean and had only the job to do to ensure sterility of the filtrate. So the best protection of the final membrane, and this again can only be verified by filterability trials. There may be different results. So here's another result where we were invited by a customer for performing filterability trials. And you see here, we have different filters. All are PES filters with different pre-filter, final filter porosities. And this is our Sandle 2 XLG with a capacity of, let me say here, 1,500 liters per 10 inch. And it was a custom process for a 10,000 liter batch cell culture medium. And when you, based on these examples here, count the number of cartridges being required for the process, it was 42 for this cartridge, it was 17 for that one, and it was only 7 for that one. So you see, it's enormous savings possible just by selecting and choosing the best filter or filter combination in the process. So here again, 2 examples. One with a culture media where the finer pre-filter layer was outperforming all the others, and here completely opposite, where the larger pre-filter membrane outperformed all the others. So take a long story short, you have to perform those trials, and afterwards you will get significant, you will see the significant differences of the different filter types, and this will can be used for optimizing the filtration process. The same for additional pre-filter. As I mentioned, you can use a heterogeneous sterilizing grade filter, and when you have a high particle burden, it makes sense to use an additional pre-filter. And this can also be tested with this kind of setup here, and there are results where you can increase the total capacity by a factor of 5 just by adding an additional pre-filter. And this cost-wise is really beneficial and makes sense to invest in an additional pre-filter because the total filtration cost goes down significantly. Okay, what is sterility? Sterility is the absence of viable microorganisms. And there are guidelines like the Annex 1 telling you that where the product cannot be sterilized in the final container, solutions or liquid can be sterilized through a sterile filter of nominal pore size and so on. So it's clear the membrane filtration technology is accepted by regulatory authorities. But all sterilization processes should be validated. And what I have explained to you before is just that water-based and in ideal scenario what is happening in the lab. But for your specific process, this has to be verified again. So sterile filtration is a validated method for sterilization. Sterility is defined as to retain 10 to the power 7 colony-forming units per square centimeter effective filtration area of a specific bacteria, which is Brevonimonas diminuta. This is how such a test is looking like, and you know this picture already, and only when it is assured that not a single bacterium has a chance to enter the liquid, then we are allowed to call it sterilizing grade filter. And this is a work that has to be done during this kind of process validation to really show that even under your specific process conditions, this will work. And there are examples for challenging liquids to be filtered. So emulsions, liposomal formulations, pack-containing formulations. This may lead to a situation where the bacterial challenge test will fail, where some bacteria will have a chance to enter the final container. And how to solve this? So you can either choose different process parameters, like the delta P. You can use alternative filter material. You can use filters with tighter pores, or also use double sterile filtration. So 2 times 0.2 micron filters. This is like that one here. So a double sterile filtration, you not only use 1, but you use 2 filters. And finally, you have to validate it that you have 0 colony-forming units afterwards. Redundant sterile filtration is different, so it's just as a kind of police filter, where you use a second filter just to avoid the situation that when one filter fails the integrity test, that you can discard your whole batch. So it's just a kind of safety function. And depending on your strategy of risk mitigation, you can decide to use a sterilizing grade filter, 0.2 micron rated, a mycoplasma retentive filter, 0.1, or even a virus retentive filter, 0.02. But don't forget that always the flow rate goes down but the safety goes up. So finally, it's a risk mitigation approach the customer and the user, the final user, has to do. So coming to the end of this talk, what about integrity testing? Integrity testing, of course, is enormously important because you will not have a chance to see all the membrane defects. So once a filter, for example, has fallen down to the floor, nobody will be able to tell you if the membrane is still intact, yes or no. So you have to fulfill regulatory requirements. You have to determine the presence of oversized pores within the membrane. You have to confirm the integrity out of the box prior and after use. And this not only detects membrane defects, but also tests the complete system. So also the housings, the tubes, O-rings, etc. are tested. So you get a higher degree of safety. And finally, prevent damage to the product by the use of defective filters. But moreover, you want also to prevent any risk to the environment. For example, when contamination by harmful bacteria may happen to the operator. This, of course, must not happen. Because time is running, I want to skip this slide. But how is this done? The integrity test is done just by increasing the pressure and through the wetted membrane, and you will then see a certain pressure decay. This will be detected by the instrument, by the integrity tester. This is correlated to the bacteria challenge test that I just explained. And depending on the technology which is used, it's called a diffusion test, or here, when you increase the pressure, it's called the bubble point test. So, but what is really important to know? Just let me skip this slide here. What is important to know? First of all, when we are talking about pre-use and post-use testing. So the pre-use test excludes potential filter damage after sterilization. It verifies the filter condition, checks the installation, and avoids the financial risk. The post-use test is really the most important fact is patient safety. You can exclude that a defect filter was used that may potentially lead to contamination of your vials, that finally the patient will be injected. So there's a big difference between the pre-use and post-use testing. And currently, there's a big discussion because this PUPSIT discussion, pre-use, post-sterilization integrity testing, is still ongoing. The background is simply there are some anecdotal explanations that say once a filter shows a membrane defect, this membrane defect may be healed during the filtration process when there are particles and clogging the hole in the membrane. We do not know this kind of situation, but currently many users have already implemented the pre-use testing because it's a GMP requirement. They have implemented PUPSIT as a risk mitigation, and there are also local differences. So in Europe, it's more prominent compared to the US. But very importantly, all filter manufacturers, they do not see increased patient safety by PUPSITing. And why is it so complex? Because you need more equipment, especially for single-use processes. You need additional filters, additional bags, and so on. So it's a really burden for the operators to do pre-use testing. But nevertheless, we have the regulatory requirements, which is here, for example, the Annex 1 telling us the filter should be verified before use and should be immediately confirmed after use. There is a new version of the Annex 1 coming out very soon, so it's expected later this year. But the story, the basic story is the same. So the wording has not changed. So anyhow, I think we have to be prepared in a way that pre-use, post-sterilization integrity testing will be seen as a must in future also in Europe. There are some nice things, like it is recognized that for small batch sizes, this may not be possible, but there's no clear definition what a small batch size is. So a lot of discussion right now, and hopefully the final version will give some final clarification for that. So the FDA in the US, it's seen differently. So here it's different wording. So that's the reason still why we have less requests from the US compared to Europe when it comes to pre-use testing. That's finally the trend that we see. There's the pre-use integrity testing is really something that's ongoing right now. Process optimization, another trend. I explained everything with the different materials. Single-use manufacturing is an ongoing trend, of course. More and more, we are also requested to get to talk about automation, because this manual operation of complex single-use assemblies is more and more complex. Continuous manufacturing, of course, is a buzzword, and large-scale single-use solutions, there are some limitations in the market right now. So the trend from filter capsules to this kind of assemblies, including sensor technology, to complete single-use operations, I think this is really the trend we currently see, and there's a big movement into innovative new solutions we would see in the upcoming years. Due to time reasons, I want to finalize with a summary. So I explained to you what the basics of filtration are: fleeces versus membrane filters, single layer versus double layer, filter formats, filtration costs, filter selection, process optimization. I explained some challenges for sterile filtration, what could be done with this kind of redundant and double sterile filtration. And at the end of this talk, we touched the filter integrity testing. But of course, each of the topic is so detailed and so much information can be given that it's not enough time to go into this kind of detail for every point. I think I exceeded over already the time. Therefore, I want to say thank you for all of you for participating. In case that there are questions, please feel free to send these questions and to ask them. Thank you very much.